Hi again, this is the second part of the revision. I'll be sharing the screen as usual. Okay, slideshow. Stopping sharing and then resharing. Let's make sure first it's recording. Yes, it is. Then let's go. Okay, this slide, as you see, shows you a very important curve, or actually two curves on the same graph that are explaining the effect of enzymes on activation energy. We have mentioned the definition of activation energy in part 1, and we said that the activation energy is the minimum amount of energy that is needed to start a chemical reaction, and that enzymes speed up chemical reactions by lowering the activation energy, meaning that they lower the amount of energy that is needed to start the chemical reaction. If we look here at the graph, actually we have a purple graph and a green graph, and they merge in their terminal part. When we look at those two arrows, we find that the green curve represents the activation energy when the reaction is catalyzed, meaning the activation energy when we are using an enzyme, whereas the purple curve represents the chemical reaction when we don't use an enzyme. This is what we call an uncatalyzed reaction. An uncatalyzed reaction means that we have no enzyme and the reaction is proceeding on its own. Here we have two energy levels. We have the energy level of the reactants and we have the energy level of the products. On the x-axis, we have reaction progress, which means the time that passes. And on the y-axis, we have Gibbs-free energy. You can ignore Gibbs-free and just say energy. Of course, we have two levels of energy here. We have the level of the reactants and the level of the products. And it's obvious that the Y of the reactants, or the energy value of the reactants, is higher than the energy value of the products. So for example, if the energy content that is stored in the bonds of the reactants is 100, and that of the product is only 30, then this 70 difference in energy will be released to the environment. And that's why whenever you find in a graph that at the beginning of the reaction here, close to the y-axis, the reactants had a higher energy level than the products, then definitely the difference has been released, and this is an exothermic reaction. So this is the first question you can, or actually the second question. The first question, what does this graph represent? This graph represents actually many things. First of all, it represents the relationship between time, or the reaction progress, and the change in energy. Second, it represents the difference in energy between the reactants and the products. Third, it represents the activation energy. Here, look, even when the reaction is uncatalyzed in the purple curve, you need to give the reactants some energy, an amount, a quantity of energy. So the energy level of the reactants, or the energy content, rises until it reaches a peak. And then at that peak, the reaction starts and we start getting the products, and the energy moves down again or diminishes again. So whether you are using an enzyme, in the green curve, or you are not using an enzyme in the purple curve, you need to provide a certain amount of energy to get this peak where the substrate is at the top of excitation and activation and would start giving the products. But there is a big difference between the uncatalyzed reaction, where the activation energy is very high, and the catalyzed reaction when we use an enzyme and the activation energy is much lower, because the peak of the green curve is lower than the peak of the purple curve. We will move on to another concept. We already talked about mechanical digestion, but here we can add a piece of information. We mentioned that we have mechanical digestion in the mouth because of the teeth. When you chew food and when you cut food, you apply physical force to cut big food particles into smaller ones. Why are we doing this? To increase the surface area. Why do we want to increase the surface area? To allow penetration of enzymes. And why do we want to allow penetration of enzymes? To facilitate the function of enzymes, to facilitate chemical digestion and hydrolysis. But the mouth is not the only place when we have mechanical digestion. We also have peristalsis. Actually, as we said before, peristalsis is present all down the alimentary tract, but it is most exaggerated in the stomach. Why does the stomach carry out the strongest peristalsis ever in the alimentary tract? Because the stomach has a strong muscular wall, stronger than that of the esophagus and stronger than that of the small intestines and the colon. Why then? What's the justification for this power to perform or carry out strong peristalsis? Because the wall of the stomach is made of 3 layers of muscles, not only of 2 layers, like in the esophagus, for example. We have 3 layers, and we will see them on the slides. Strong peristalsis in the mouth, strong peristalsis in the stomach or in the gastric wall is called churning. So what is churning? Churning is a mixer-like effect. It's like when you churn food and liquid in the mixer to get a juice, for example, or to make soup. So you're mixing the particles together and further particulating the food substances. So what's the importance of mechanical digestion and what's the importance of churning? Because churning is one powerful form of mechanical digestion. Number one, when the stomach churns food, there is further particulation of food, meaning that we further break down the big food particles into smaller ones. Number two, the stomach, upon churning food, will mix the food particles with the gastric juice, with the components of the gastric juice, and therefore will help the process of digestion. Now, chemical digestion involves one type of chemical reaction, which is hydrolysis. And as we said before, we use water to break down larger molecules into smaller ones. Here we have a good example and a famous example. Why are we focusing on that example? Because this example shows you here a molecule that is called sucrose. You needn't know the chemical formula of sucrose, but you need to identify it. When you see the molecule, you can understand that this is sucrose, and you can interpret or explain what's happening here. So we have 2 cyclic molecules or ring-like molecules that are bonded together through this covalent bond that is called the glycosidic bond. You needn't know the term. So this is a disaccharide. So sucrose, which is common table sugar, is considered a disaccharide. Why is it considered a disaccharide? Because it consists of 2 simpler monomers, which are the monosaccharides, glucose and fructose. Glucose and fructose. So what we are doing here in this diagram, if you are ever asked to explain or interpret what happened or justify why we could separate the 2 monomers from each other, actually, we added water. We added H to one of the 2 monomers and OH to the other monomer. So we split them apart. This is a process of hydrolysis. Hydrolysis is here in hydrolysis of sucrose, we broke down sucrose into the 2 monomers, glucose and fructose, by the addition of water. So you might get the diagram, or I might ask you directly about the results of hydrolysis or digestion of sucrose. Sucrose is table sugar, sucrose is cane sugar, and sucrose consists of two monomers, glucose and fructose. So the final products of digestion of sucrose are two monosaccharides, which are glucose and fructose. And as we mentioned in worksheet 11, glucose monomer is present in all disaccharides and all polysaccharides. Now we come to the teeth, which carry out mechanical digestion. Of course, you have to know the types of teeth. We have, in general, we have three types of teeth: the incisors, the canines, the premolars, and molars, actually four types. The incisors and the canines are common between milk teeth and permanent teeth. Similarly, the molars are present in milk teeth and permanent teeth. Only premolars differ because premolars are not present in milk teeth. We cannot see premolars in a set of milk teeth. The number of incisors is the same in milk and permanent teeth. The number of canines is the same. So we have 4 upper incisors and 4 lower incisors, 2 central and 2 lateral. And then we have 2 canines on each side of the incisors. This is common, this is the same in milk teeth and in permanent teeth. The incisors have sharp, broad edges in order to cut and bite food. The canines have pointed edges in order to tear food, in particularly meat or flesh. The premolars are totally absent in milk teeth. In permanent teeth, you have premolars. You have 2 premolars on each side. And then you have 3 molars on each side. So all in all, each of the jaws, the upper jaw and the lower jaw in permanent teeth, each of them contains 16 teeth. So if I ever ask you to compare milk teeth and permanent teeth, first of all, you would compare and contrast sorry, the number. The milk teeth are 20 in number, whereas the permanent teeth are 32, or you can say 10 per jaw and 16 per jaw. Milk teeth are whiter. Milk teeth are shed at a certain age. They are deciduous, and that's why we sometimes call them deciduous teeth, whereas permanent teeth are not supposed to be lost unless you have very bad hygiene, dental caries, and root affection of the teeth. In milk teeth, there are no premolars at all, whereas in permanent teeth, we have 2 premolars on each side per jaw. And concerning the molars, they are present in both, but milk teeth have 2 on each side per jaw, whereas permanent teeth have 3 on each side per jaw. You needn't know the timing or the time at which the tooth erupts, the permanent tooth erupts. And then we come to the tongue. Of course, we have several. I'll remind you of the tissues that are present in the tongue. The tongue can move during eating and during speaking because it contains muscular tissue. So one of the functions of the tongue is that it helps in speaking. And the other function is that it helps in moving food inside the mouth for chewing by the teeth. All this is because of the muscular tissue. And the muscles in the tongue, of course, as you know, are voluntary. You can give voluntary orders upon request to your tongue to speak, or to your tongue to articulate words, or to help in moving food inside the mouth cavity. But the tongue also contains lingual tonsils. There are scattered patches of immune tissue in the tongue for defensive mechanisms against microbes, but most importantly, it contains nerve endings. And these nerve endings are called taste receptors, and we have every site in the tongue is most specialized in a particular taste. So at the tip or anterior most part of the tongue, we can best taste sweet. At the back part, we can best taste bitter. On the lateral aspects and anteriorly or to the front, this is salty taste. And then laterally on each side and posteriorly, we can taste sour. And umami taste is all over the tongue, and umami taste is that pleasant sensation that causes or triggers secretion of saliva. There are certain types of food that are famous for triggering secretion of saliva, like, for example, spices, like soup, like fermented cheese, and and monoglutamate sodium as a preservative. Monoglutamate sodium is a preservative that is added to food to prolong the shelf life, so that if you keep the preserved meat outside the fridge in cans on shelves in a supermarket, then it wouldn't get rotten. But unfortunately, monoglutamate is carcinogenic, meaning that it can stimulate and trigger the occurrence of cancer colon. However, monoglutamate sodium stimulates this umami taste, stimulates salivation, formation of saliva, and that's why people add it not only as a preservative, but also to stimulate this umami pleasant sensation. Concerning digestion in the mouth, we have two types of digestion in the mouth. If I ever ask you what kind of digestion can occur in the mouth, you would say mechanical and chemical. Of course, mechanical digestion is what is carried out by the teeth, whereas in the form of chewing, whereas chemical digestion is through enzymes. And the only enzyme that is present in saliva is salivary amylase. So salivary amylase, as its name indicates, is secreted by the salivary glands. It is present in saliva. And if I ever ask you to define or to explain or describe the function of salivary amylase, you would say, as usual, whenever you are required to describe the action of any enzyme, you would say it catalyzes the hydrolysis of starch, of the polysaccharide starch, into the disaccharide maltose. What's the optimum pH of salivary amylase? It's the pH of saliva. Type, what's the pH of saliva? It's slightly alkaline. It is 7.2 to 7.4. What's the reason behind the alkalinity of the saliva, and therefore that the optimum pH of amylase is salivary amylase is weakly alkaline? The presence of sodium bicarbonate. Why is there sodium bicarbonate in saliva? In order to protect this we didn't mention, but we will be mentioning it in the presentation on infection control, because bacteria ferment the remains of food in your mouth cavity and produce lactic acid. That lactic acid can dissolve calcium of the enamel of your teeth, and therefore there should be sodium bicarbonate to neutralize and buffer that acid. This leads us to the composition of saliva. What does saliva consist of? Saliva is a digestive juice, and therefore, like any other digestive juice, it consists of 90% water. And water is needed for what? Water is needed for hydrolysis. What else is present in saliva? We have just mentioned, other than water, we have mentioned sodium bicarbonate. If I ever ask you to mention the importance of sodium bicarbonate in saliva, you will just say provide the optimum alkaline pH for action of salivary amylase enzyme. And then we have a slight mucus in saliva in order to make food sticky, to make the particles sticky together, and the bolus of food easily swallowable. Don't mention anything about coating, like in the stomach. So whenever you have a question about mucus, you have to be very careful. Am I asking about the function of mucus in the mouth cavity or in the stomach cavity? In the mouth cavity, it's only lubrication of food, sticking food particles together, facilitating swallowing. What's the only food stuff? Another question. What's the only food stuff that is tackled in the mouth, that is digested in the mouth? It's starch. Is it a carbohydrate or a protein or a fat? It is a carbohydrate. Is it a polysaccharide or disaccharide or monosaccharide? Starch is a polysaccharide. Okay. Can we start digestion of glycogen, which is also a polysaccharide, in the mouth cavity? No, Why? Because glycogen is a more complicated and complex carbohydrate polysaccharide actually than starch. So it needs a more effective enzyme. The more effective enzyme is pancreatic amylase. Pancreatic amylase is secreted by the pancreas and works in the small intestines. Here, this is an important slide where we have three polysaccharides: starch, cellulose, and glycogen. And they all contain maltose. So if starch is first digested in the mouth, it would give the disaccharide maltose. Can starch be completely digested into the monosaccharide glucose in the mouth cavity? No. So the digestion of starch that occurs in the mouth cavity is an incomplete digestion. Starch is the storage form of carbohydrates in plants. That's why when we eat potatoes or sweet potatoes or any kind of plants that contain energy reserves, then it would be starch. Glycogen, on the other hand, is another polysaccharide, but as you see, it is more branched and more complicated than starch, and it needs a more developed enzyme, a more elaborated enzyme, which is pancreatic amylase. So glycogen is not at all digested in the mouth cavity, although it is a polysaccharide like starch, but because it is more complicated. Cellulose is a polysaccharide, and it is made of the same components of units of maltose connected together. But the problem is that, first of all, let me tell you where cellulose is present. Cellulose is present in plant cell walls. So it's not, yani starch and cellulose are both present in plants, but they are different. Starch is mainly a storage form. When the plant produces excess glucose, then it will store that excess glucose in an insoluble form called starch. But cellulose is a polysaccharide, yes, but it is only present in the wall surrounding the plant cell in order to protect it and give it shape. When you eat any kind of plant with its skin, potatoes that are unpeeled or cucumbers or whatever thing that is unpeeled, you're eating the cell walls actually, and you're eating cellulose. These cellulose fibers are not digested in the human small intestine, so cellulose is not digested. We don't have the enzyme cellulase that digests cellulose. Rabbits do, cows do, buffaloes do, all herbivorous animals do, but we do not have cellulase enzyme. Now, as you see, the pharynx, we explained the pharynx before, but let me tell you here that the pharynx is a common passageway for food that should be directed to the esophagus and air coming from the nose. So if you are inhaling air, if you're getting in air, air will pass from the surrounding to the inside of the nasal cavity, and then to the nasopharynx, to the oropharynx, and then to the laryngopharynx, and then from the laryngopharynx to the larynx itself and the trachea. In such a case, the uvula should be hanging down, as you see, and the epiglottis should be upright in order to allow air to pass. This is exactly the opposite of what happens during swallowing. We described the function of the epiglottis before. It contracts and moves downwards to seal the glottis, and then this results in pushing of the food or forcing the food to go to the esophagus. But when you are inhaling air, the epiglottis should be upright in order to allow air to enter the trachea. And now we come to the esophagus. The esophagus is the link between the lowermost part of the pharynx, which is the laryngopharynx, and the stomach. So the esophagus is connected upwards to the laryngopharynx, and it is connected downwards to the cardia of the stomach, to the first part of the stomach. As you see, the esophagus passes through the neck and then passes through the chest cavity behind the heart and in between the two lungs, and then penetrates the diaphragm through the hiatus, as you know, the opening in the diaphragm, in order to reach the stomach. The wall of the esophagus is muscular. In the upper third, it's striated. In the lower third, it is totally unstriated. And in the middle, it's a mixture of the two. But if I ever ask you, you just say unstriated, involuntary, smooth muscles, because the 2/3, the majority of the esophagus, are smooth, unstriated, involuntary. And it is formed of two layers of muscles, the circular layer, an inner circular ring-like layer, and an outer longitudinal layer. So what's the function of the smooth muscles of the esophagus? Peristalsis. And what's peristalsis? It's a series of regular contractions and relaxations of the muscular wall of the esophagus. Is it confined to the esophagus? Is it restricted to the esophagus? No, it's all down the alimentary tract. What's the function? Mainly peristalsis in general helps sweeping down food, helps pushing food down the alimentary tract. But peristalsis in the stomach has a different aspect because the wall of the stomach consists of three layers: an outer longitudinal layer, a middle circular layer, and an inner oblique layer. It is as if we added only the inner oblique layer. If you open a window in the wall of the stomach, you will find those folds. These are called the rugi. So if I ever ask you, what are these? These are folds in the inner gastric mucosa. How do we call these folds? We call them rugi. Why are they important? Because they allow distension of the stomach with food. What's the type of muscles in the wall? The layers have been mentioned: outer longitudinal, middle circular, and inner oblique. But what about the type of muscles? It's the same type of muscle as in the esophagus. The wall of the stomach consists of smooth, unstriated, involuntary, smooth muscles. What are the parts of the stomach? This part that is close to the end of the esophagus is called the cardia, and then that convex upper part is called the fundus. That lowermost part is called the pylorus, and before the pylorus we have a white reception that is called the antrum, and the rest is called the body of the stomach. The main bulk of the organ is called the body of the stomach. This is the outer curvature, and this is the inner. Inner means towards the middle line of the body. Where does the esophagus lie? The esophagus lies in the upper left quadrant of the abdominal cavity. What do these arrows represent? They represent the direction of movement of the foot. So here, those arrows that are arranged in a circular fashion represent churning. You see the direction of churning? It's circular, like the mixer effect. It's not downwards like peristalsis, but the idea is the same. Churning is nothing but strong peristalsis, and peristalsis is nothing but powerful contractions and relaxations. Here we have a very important structure. I wonder whether it's on the slides or not. Yes. Here we have a very important structure that is called the cardiac sphincter. What is a sphincter, and what is the cardiac sphincter in particular? A sphincter is a ring of strong involuntary circular muscle fibers. So it is a strong ring of circular muscle fibers. It cannot be longitudinal. It cannot be oblique, although the rest of the stomach contains such muscles. But the sphincters, whether we are talking about the cardiac sphincter above or the pyloric sphincter below, they are strong rings of muscles that can contract forcibly and relax easily. So what's the role of the cardiac sphincter? When the cardiac sphincter is relaxed, when the muscles of the cardiac sphincter are relaxed, are not contracted, they allow passage of food from the esophagus to the stomach. But when they contract, the sphincter constricts, then this prevents regurgitation of food into the esophagus. This is the cardiac sphincter. Concerning the pyloric sphincter, the difference is a bit in the principle, the fundamental function is the same, contracts and relax, but there are minor differences. So when the pyloric sphincter contracts here, it retains food inside the stomach. It keeps the food long enough inside the stomach cavity in order to give enough time for proteins to be digested. Because as you know, the first time proteins are hydrolyzed is in the stomach cavity, not in the mouth cavity. But when the pylorus or when the pyloric sphincter relaxes, when it dilates, when the diameter increases, then food will pass to the duodenum. I might ask you about the dilatation of the pyloric sphincter, the relaxation of the pyloric sphincter. Does it dilate continuously? Does it relax continuously? No, because if it opens all of a sudden and it opens continuously, then this would lead to food passing all of a sudden from, or chyme passing all of a sudden from the stomach to the duodenum, and this would be a burden on the duodenum, and there would be maldigestion or bad digestion. So it opens and closes at intermittent intervals. The difference between, let's first see what the gastric juice consists of. The gastric juice, just like any digestive juice, consists of 90% water for hydrolysis, which is the breakdown of food molecules by the addition of water, and it contains hydrochloric acid. Hydrochloric acid is there, although it's a risk factor because it's acidic and can cause eating up of the gastric mucosa, yet it is important because it kills the germs in food. So HCl is important because it kills the germs in food, and by being there, it will provide a very strong acidic pH to the stomach, meaning that the pH of the stomach will be very low. And actually, because we have a lot of HCl in the stomach, then the pH of the stomach is 1.5 to 2.5. And since pepsin is there accompanying HCl, then the optimum pH of pepsin should be acidic. So if I ever ask you about the optimum pH of pepsin and you tell me 1.5 to 2.5, I would ask you, is it acidic or neutral or alkaline? You would say it is acidic. Is it strongly acidic or weakly acidic? It is strongly acidic because it's far below 7. What's the cause of this strong acidity and this very low pH in the stomach? HCl. Why is HCl there? To kill germs in food, to provide the optimum acidic pH for action of pepsin, and to first activate pepsin, because we need to activate pepsin first. So we say activate pepsin before saying providing optimum pH. Why do we need to activate pepsin? We need to activate pepsin because pepsin is secreted in the form of a proenzyme, which is the least active form. Actually, the proenzyme of pepsin is called pepsinogen. So pepsinogen is the proenzyme of pepsin. It is inactive. How can we convert it into an apoenzyme? By hydrolyzing it, by adding water to break part of it. When we break part of the proenzyme, actually we are breaking the part that covers, that hides the active site. So when this part is removed, the active site is now obvious and seen and exposed. But are we now active enough to be able to catalyze digestion of pepsin, of proteins? Not yet. This is still an apoenzyme. So an apoenzyme is a proenzyme that has been hydrolyzed to expose the active site. When we get the apoenzyme, it still needs to be combined to an apoenzyme or a proenzyme to be converted into a hollow enzyme. So the hollow enzyme is the most active form of pepsin. What's the action of pepsin? Pepsin catalyzes the hydrolysis of pepsin into short molecules or peptides, short peptides. Is this the final form of protein digestion? Is this the final simplest molecule of protein digestion? No, we still need to be converted into amino acids. So is digestion in the stomach partial or complete? Digestion in the stomach is partial, because we still need to further hydrolyze the peptides into amino acids. Where are we going to obtain all these final products of hydrolysis? Where are we going to obtain all these simple soluble products? in the small intestine. Are pepsin and HCl useful? Yes, they are. HCl function, we have just mentioned, and pepsin is important to hydrolyze proteins, but they are double weapons because they constitute risk factors. They represent risk factors. Why? And pepsin, being a proteolytic enzyme, it can digest the proteins in the gastric mucosa. HCl, being an acid, can erode or it's a corrosive substance actually, so it can eat up the cells of the gastric mucosa, all in all, proteins, carbohydrates, and fats. So how are we going to protect the stomach wall? By secreting mucus. So the gastric mucosa contains mucus secreting cells that are protective. So mucus is a protective factor. What kind of protection is it? It is a mechanical, physical kind of protection, because mucus, being thick and being sticky, will coat the inner lining of the stomach and act as a mechanical or physical barrier between HCl and pepsin in the center of the stomach, in the lumen of the stomach, and between the protein-containing cells of the gastric mucosa. So if for any reason HCl increases or mucus decreases, then you have an increased risk factor or a decreased protection. Risk factor for what? And decreased protection against what? Against peptic ulcer or gastric ulcer. Gastric ulcer is, I'll show you gastric ulcer. Peptic ulcer is a general name for gastric and duodenal ulcers, and this is how you can see the ulcer when you use an endoscope with a camera, with a video camera, to visualize the inside. You will find like a hole in or a depression in the mucosa of the stomach. This is because here there were gastric cells that have been eaten up by pepsin and HCl because of decreased protection. And around them, you have a very dark red area resulting from increased blood flow to the area because of inflammation. So this picture represents peptic ulceration. Which people are more likely to get peptic ulcer? People who have increased HCl secretion, people who are all the time under stress, people who are heavy smokers, people who consume large amounts of alcohol, people who take analgesics on an empty stomach, people who take cortisone or steroids in general because they increase HCl, people who are nervous, or people who are congenitally having a diminished mucus secretion. And there is a last factor which is very important. It's last but not least, the presence of H. pylori bacteria. Why are H. pylori bacteria causing peptic ulcer? Because H. pylori bacteria can destroy the mucus secreting cells. If you destroy the cells that secrete mucus, then you will not have enough mucus to protect the stomach. And as I told you in the classroom, and this was revolutionary in the treatment of peptic ulcers. In the past, we used to treat peptic or gastric ulcers using antacids, meaning that the patient has increased HCl secretion, then we will give him something to diminish HCl secretion, or something that is milk-like and coats a suspension that coats the inner gastric mucosa. But after H. pylori bacteria have been bacteria have been identified as a cause for peptic ulceration, the treatment is now through antibiotics. Now we have two other diseases other than gastric ulcer that are related to the stomach. The first one is due to a weak cardiac sphincter. When you have a weak cardiac sphincter, then if you overeat and your stomach is very distended and the sphincter gets distended because it's weak, or if you eat a heavy meal and then lie down on the spot and get recumbent on the spot, then some acid will go upward in your esophagus, and that shouldn't occur because the esophagus is not as protected as the stomach. We call this esophageal reflux disease, or GERD. G stands for gastro, E stands for esophageal, R stands for reflux, and D stands for disease. Reflux means upward flow or backflow in the wrong direction. The causes, of course, are essentially a weak cardiac sphincter, or you might have an almost normal cardiac sphincter, but you have increased gastric acidity for the reasons we have mentioned before. What would the patient suffer from? The patient would suffer, of course, from heartburn and maybe pain, esophageal pain, because as I told you, when the acid goes upwards in the wrong way to the esophagus doesn't have, it's true that the esophagus secretes mucus, but it does not have the same coating as the stomach, and therefore there could be some inflammation in the inner wall of the esophagus, or there could be ulcers, and this would lead to esophageal pain that is sometimes confused with or misled for misdiagnosed as being cardiac disease or cardiac pain. On the other hand, we have hiatus hernia. Hiatus is the normal opening in the diaphragm that allows the passage of the esophagus. If that hiatus is of normal size, then no problem would occur. It will just allow the esophagus to go from the chest cavity to the abdominal cavity and join the stomach. But if for a reason or another that hiatus, that opening in the diaphragm, is wider than normal, then part of the stomach will slip upward. Part of the stomach will herniate. To herniate, hernia means bulging in the wrong place. So the stomach will slip upward in the chest cavity. The stomach is not supposed to be here. So first of all, the stomach will be compressed at its waist, and this causes gastric pain. Food will be stagnant here, and this will result in bloating. So you will have pain. You will have bloating and gases. You will have heartburn. The cardiac sphincter is no more supported, and it gets wider, and you might have dyspnea or difficult breathing because of compression of the lungs by the bulging stomach. Chyme, of course, is nothing but the contents of the stomach. Chyme is heavy, semi-solid. It's neither fluid in consistency nor solid. It consists of the components of the stomach that have been churned and exposed to the churning mixer-like effect of the stomach. Of course, the chyme contains, of course, water. It contains HCl. It contains partially digested food. It contains vitamins and all what you have ingested. When this chyme falls into the duodenum, then it will stimulate the secretion of bile. The liver is the largest organ in the body, and if the stomach lies in the left upper quadrant, the liver lies in the right upper quadrant of the abdominal cavity, and the liver has many functions: storage of excess glucose as glycogen, converting toxic substances into non-toxic ones, which we call detoxification, and most importantly, and most related to the process of digestion, is the secretion of bile for digestion of fat. We always say, here, of course, you can see the liver and you can see the gallbladder underneath. And we always say that the bile is secreted by the liver cells, but concentrated and stored in the gallbladder. And the final release of bile into the duodenal cavity is a function of the gallbladder, because the wall of the gallbladder is muscular. It consists of smooth unstriated muscle fibers, and therefore when it contracts, it releases the concentrated bile into the duodenum. What's the trigger for secretion of bile into the duodenum? The presence of fat in the duodenum, because bile helps digestion of fat. We always say that bile is a digestive fluid, but it is a non-enzymatic digestive fluid. Why digestive? Because it helps in the digestion of fat. Why non-enzymatic? Because it does not contain any enzymes. So if bile helps digestion but does not contain any enzymes, then definitely bile cannot carry out chemical digestion. It only carries out mechanical digestion, a process that we call emulsification. What is emulsification? Emulsification, I don't know whether we have a picture for this or not. Emulsification is converting the big fat globules into smaller fat droplets in order, yes, we have the picture here. We have those fat molecules, and here you can see that we divided them into, but this is not, I'm sorry, this is not emulsification. This is chemical digestion by pancreatic lipase, because this is at the level of the molecules. Emulsification, the diagram of emulsification would be like the diagram of mechanical digestion, a big fat globule that is divided into smaller ones. Here we have the fat molecules, and they are digested into fatty acid and glycerol. So we are working at the level of molecules. So this is chemical digestion. And the enzyme that catalyzes hydrolysis of fats into fatty acids and glycerol is the pancreatic lipase enzyme. Actually, the juice of the pancreas consists of 90% water, sodium bicarbonate. And sodium bicarbonate in the pancreatic juice is very important, because this pancreatic juice will drop into or will fall into the duodenum. So the duodenum also receives chyme. So there is chyme coming from the stomach, and there is pancreatic juice coming from the pancreas, and they'll both meet in the duodenum. Chyme is strongly acidic, and the small intestines is not prepared to receive such an acidic juice. That's why the pancreatic lipase with its sodium bicarbonate helps neutralize the acidity of chyme first, and then provide the optimum alkaline pH for action of all the pancreatic and intestinal enzymes that work in the duodenum. Because as you know, the duodenum receives chyme from the stomach, the pancreatic juice from the pancreas, as well as the juice that the duodenum or the intestines themselves secrete. So all the pancreatic enzymes and all the intestinal enzymes that work in the duodenum love the alkaline pH, which is strongly alkaline, from 7.5 to 8.5, and this is the function of sodium bicarbonate. Of course, here you see the small intestines, and you can see that the longest part is the terminal part, which is the ileum. The small intestine is where the final products of digestion are obtained. It contains 90% water, it contains sodium bicarbonate, it contains mucus for protection, it contains peptidases for final digestion of the peptides into amino acids. So what's the function of peptidases? To catalyze the hydrolysis of peptides into amino acids. Therefore, what are the final products of protein digestion? The amino acids. Therefore, where are the final products of digestion of proteins? The amino acids obtained in the cavity of the small intestine. The intestinal juice also contains disaccharidases to digest disaccharides or to catalyze. Of course, if this is exceeding slide 71, then it wouldn't, you wouldn't be asked about it unless through a comprehension passage. Disaccharidases catalyze the hydrolysis of disaccharides into monosaccharides. And here we can see examples. Let me see where we are, on which slide we are, sorry. Let me check first we are still recording, because it would be catastrophic if we're not recording. Yes, we are. Which slide is this? Let me move on. Yes, this is slide 71. So the slides on disaccharides and disaccaridases are not, you are not supposed to know them unless unless you have a diagram or you have a passage. I'm going to share the screen, I'm sorry. I should have converted it into a slideshow. I'm not focused. Right, let me stop sharing to make sure that we are on the right path. Yes, we are. Okay, sorry for the delay. So these slides you just have to read to be prepared for the comprehension passages. So sucrose is hydrolyzed into glucose and fructose. Lactose, which is milk sugar, is hydrolyzed into glucose and galactose, and maltose into glucose and glucose. Let's move on and see what's there. The three enzymes, sucrase, lactase, and maltase, this sucrase corresponds to sucrose, so it catalyzes the hydrolysis of sucrose into glucose and fructose. Lactase catalyzes the hydrolysis of lactose into glucose and galactose, and maltase catalyzes the hydrolysis of maltose into glucose and glucose. Notice that the only difference between the name of the enzyme and the name of the sugar is that the enzyme has an A instead of an O. And therefore, if we review the reactions, we can see that the final products of digestion of carbohydrates, which are the monosaccharides, are all obtained in the cavity of the small intestine. This is number one. And number two, that the monosaccharide glucose is common to all the disaccharides. All the disaccharides contain glucose. As I told you, the large intestine you will only be asked about it through a comprehension passage. Thank you guys and wish you the best of luck tomorrow, inshallah. Bye.